Hello everyone and welcome to today's live broadcast, How to Deploy a Portable Lab for the Surveillance of Emerging Infectious Diseases, presented by Josh Quick, a research fellow at University of Birmingham. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Oxford Nanopore Technologies. Oxford Nanopore Technologies aims to disrupt the paradigm of biological analysis. Their technologies and commercial model has already opened up DNA analysis to researchers who previously had no direct access to sequencing technologies, freeing them up to perform analysis in their own labs or in their field or in real time. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type that question into the box that appears in the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, notice that you will be viewing this presentation in a slide window. To enlarge that slide window, click on the screen icon located at the lower right. If you have trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. This presentation is educational and thus offers continuing educational credits. Please click on the button in the bottom left corner and follow that process for obtaining your credits. Without further ado, please join me in welcoming Josh Quick. I will now turn the presentation over to him. Welcome, sir.
Thank you very much for the introduction, Susie. So thank you for that introduction. My name is Josh Quick and I'm a researcher at the University of Birmingham. We are a genomics research group. We try to find new innovative ways and applications for genomics or DNA sequencing technologies, of which Oxford Nanopore have developed such a technology. And I have traveled to various outbreaks around the world, taking with me a mobile lab with at its heart an Oxford Nanopore portable DNA sequencer. And we have been able to use that technology to tackle and control ongoing outbreaks. In this particular talk, I'm going to be talking about two viral epidemics, which you will definitely have heard of. The first is the Ebola outbreak in West Africa, and the second one is the Zika outbreak in South America. And we used portable genomics and surveillance, we call it, which means I'm using to refer to when you know that something is there and you want to keep an eye on it rather than trying to find something new. And what we did in those countries. So the Oxford Nanopore MinION technology is very interesting because it's allowed, among other things, the first truly portable genome sequencing to be performed, and it's really opened up this whole new area of field sequencing. So previous generations of instruments were quite large, around the size of a photocopier, something like that, or maybe a laser printer, but they weren't very portable. Whereas the MinION, because of its technology, it doesn't have any moving parts. It means that it can actually connect to a laptop computer, and it's only really the size of a USB stick. So, give you some examples of some mobile sequencing. So this is us at a brewery in Austin, Texas, at the Jester King Brewery, doing some analysis on some spontaneous fermentation beers, and we're actually able to take everything down there. It's a sort of hobby of ours, really. It's more of a sort of public engagement activity, and we like to look at fermentation and what's the microbiology involved in fermentation, and do it right there and then in the field. This is an example from Aberystwyth, Arwyn Edwards, Aberystwyth University, where he's actually doing DNA sequencing in the Arctic at the 78th parallel, and he's actually collecting samples doing the analysis in a mobile lab, and then actually making a decision about what to do next, maybe move the sampling location, or give up altogether, or something like that. So that's an example of where you can improve the efficiency of working in these types of environments. This is a photo from a researcher called Aaron Pomerantz, who was using the MinION to identify species of snakes in the Ecuadorian jungle, and you can see all the equipment they used there. This is an underwater laboratory called the Aquarius, where they test out technology in sort of austere environments, where there's, this is 25 meters under the sea. And this one, you may have heard of this one in the news. This is aboard the ISS, and this is astronaut Kate Rubins, Kate Rubins, who's been testing out the use of the MinION in space, so or near space orbit. So that's an example of a sort of more future-looking project into whether or not the MinION could be used on long missions, or manned missions to Mars, and whether or not the MinION could be used to detect and diagnose infection for treating illness on those long journeys. So now to talk about my experiences. This is my mobile lab that I took to West Africa in April 2015, and this is my trolley at Heathrow Airport, about to board a plane to the capital of Guinea in West Africa, Conakry. So, how did we get to this point, and what did we have to do to put this equipment together? So I'm going to go through some of the key things. So I've already mentioned the reason that we chose the MinION is because it's a very small instrument compared to the previous generations of instruments. It's very lightweight and requires sort of fewer support equipment, so it doesn't require mains power, things like that, which makes it very good for an environment where there might be frequent power cuts. It's also relatively cheap. The actual the flow cell is actually a disposable consumable, which now allows us to analyze these samples relatively cost efficiently. So that's going to be the first thing that you pack in your mobile lab. So the device itself, this is the product. It's a small sort of hinged device with the main stuff going on in this disposable flow cell, which is a single-use flow cell. So this is the top part here. You actually, the way that it works is you connect this to your computer, you prepare what we call a DNA library, and then you drop your library onto the area here, which is where the tiny nanopores are, which is what read off the sequence of the DNA that you put onto the flow cell. And I'll go into that in a little bit more detail in the next slide. So nanopores are biological proteins, and they're ubiquitous in nature. This particular protein is a membrane protein or lipoprotein from E. coli, and pores insert into membranes, and in this case it's an insulating membrane, which means that so that if a voltage is applied to it, then ions have to flow through the middle of the pore. So you can see in this diagram here, those small dots represent ions flowing through the pore, which is the only way they can get through, and that creates if you measure the current across the membrane, that means you create the signal like this, and the actual DNA strand within the pore is causing a blocking of that ion flow, which is giving a characteristic change in the current level, which can be measured. And that DNA will travel through the pore, the speed of which is controlled by this protein here, which is we call it the motor protein, which basically slows down the DNA in order to enough that it can be read. And so that's slowing down and controlling the speed of the DNA traveling through the pore, and then it's being read out as a current level, and then it's being read off the instrument onto the laptop computer, where it's going to be converted into base calls or bases, which is obviously the alphabet that DNA is made up of. And here's another view showing the same thing, but the actual sequence that you're going through that's within the barrel of the pore is more than one base at any one time. So you can see in this diagram each coloured bar represents a base, but the pore is long enough such that there's multiple bases within the pore, which means that when you actually read off a current, you're actually reading off a word. It's actually a DNA word, because it contains more signal which is coming from other bases as well. And I've represented that here by using this pretend sequence here. If you imagine this sequence going through the pore, and if you just think about the base, the C base, you're first observing it at the last position, and as it slides through, you'll then observe it 5 times in this case, where we're assuming that the word length is 5 bases. So it's a very, very tiny measurement that you're making. It's a very small single molecule measurement that you're making, but because you get multiple opportunities to observe the base, you can use that to combine these small bits of signal into what is actually remarkably accurate DNA sequencing. So each the actual DNA, the actual sequencer is reading, or the chip underneath the flow cell is called the ASIC, and it's reading off what we call these samples, so raw samples. So it's actually sampling the current at 6,000 hertz, so 6,000 times a second. And each of these dots on this plot represents an individual sample. And you can see that you account for some variation in the samples, but the red lines represent what we call events, and those represent 1 kmer, being 1 kmer in the pore. So kmer, sorry, 1 word within the pore. So as the sequence progresses by 1 base, you can see that you end up with these signal levels. And then this can be converted by using a base caller, which means is a general term which means calling from the data that you've recorded the actual DNA sequence. And in this case, we use a base caller that's a piece of software called Albacore, and it takes these raw samples and it uses a machine learning algorithm that's been trained on known sequence to determine what the actual sequence of that read actually is. And once we have base calls, we call them reads, and that means a DNA read represents the sequence of 1 molecule, and that can be used for doing downstream analysis. So the MinION was first launched in May 2014, so it's the newest DNA sequencing technology. It's only been out less than 3 years, and it's been characterized by a very rapid progression in the technology, which is I think fairly common of any sort of groundbreaking new technology, but there's been a very rapid improvement from the first days that we got our hands on this instrument. So I've just summarized some of the developments over the months. One of the big ones was the move to a new pore, which I mentioned before. It's a pore derived from E. coli bacterium, and it replaced an earlier version of the POR, and it was found that it improved the base calling accuracy, so the ability to accurately read off the correct sequence of the DNA strand that's passing through the POR. So another unique feature of nanopore sequencing is that it reads whole molecules, which means instead of reading short defined lengths of DNA, which is how many previous technologies would have worked, it can read whole long fragments of DNA, and really it depends. It can really read anything that you can manage to get into the actual device. So this is an example of the amount of data which we're talking about here. So these are some runs that we performed when we actually sequenced the human genome as a side project on the MinION, and these numbers are the amount of bases that you can get from one flow cell. So in the best case scenario, we were getting 4 × 10⁹ bases, which is 4 gigabases, which is approximately 1 times the human genome. If you were to read the human genome once, slightly more than once. It's also equivalent to about 1,000 times reading a typical bacterial genome. And these are examples of the read lengths which you can achieve. So for a typical human run that's shown here, for different types of libraries, we were able to generate reads anywhere from about 10,000 bases, anywhere up to about 900,000 bases. So it's unique in its ability to read extremely long pieces of DNA. So what else are you going to include do we need to include in our mobile lab? Well, we devised, certainly for I'm going to talk about how we sequenced Ebola first of all. We decided that the simplest way to try and sequence Ebola would be to use a technology, an old technology called PCR, the polymerase chain reaction. And what that allows you to do, so this is the equipment that we took with us. Obviously, there was some labware in the top picture, but there was also some cold chain reagents. So most molecular biology reagents, enzymes, things like that, they require being kept chilled or frozen to extend their life. So this is not really the ideal situation, but it means carrying reagents on cold packs to keep them cold. So we use these polystyrene boxes which are insulating and pack them with cold packs to keep them as cold as possible when we're traveling. But once we had it all set up, the actual equipment that we needed to do this Ebola sequencing was only that much stuff. So not a great deal amount of stuff. It can fit on 2 tables. And the equivalent setup for a conventional DNA sequencer, and by conventional I mean a benchtop instrument, which might be the size of a laser printer, required this amount of equipment from another group that were also sequencing and helping do surveillance sequencing in Sierra Leone instead of Guinea. So PCR, you probably will have heard of the polymerase chain reaction. It's basically a technique where you start off with a small number of templates, and using small oligonucleotide primers, you generate billions of copies of that template that you're interested in. And it's notable that it's quite a robust technology, but it's also very sensitive. In theory, a single template is enough to generate billions or trillions of copies, because the reaction is exponential. For every 2 for every molecule you get 2 more, so you get an exponential number of copies. So it's very good. It's got very good sensitivity, and that's one of the reasons that we chose it for this project. So the way that we decided to do it was to design primers which would cover the entire Ebola genome. So Ebola is a small RNA virus, which means it uses RNA as its encoding its genome. It contains only 7 genes, which are represented by the blue arrows, and is only about 18,000 bases long. So what we decided to do, given its small size, was to design lots of 500 base pair amplicons, we call them, which is products generated by PCR or amplification, but 500 base pair products which go across the genome and are next to each other or slightly overlapping, such that we call it tiling our way along the genome using lots of short products. When we got there, we then realized that we could actually miss out every other primer and generate 1,000 base pair reactions, and then every fourth, and generate 2,000 base pair amplicons, and could tile across the genome in 2,000 base pair products and only use 11 reactions. So some of the early data that we generated in the UK and in Guinea showed that we were able to sequence the entire Ebola genome using these overlapping amplicons that I described. Left to right is the genome, the coordinates of the genome, so 0 to 18,000, and then the Y in Y is the number of reads that are aligned to that position of the genome. So we call that the depth. So using these 11 reactions, we were able to cover 98.4% of the Ebola genome at high depth, which meant that we were able to recover 98% of all the bases in that genome for analysis. So what to quickly say how you then go from a PCR product to the sequencer, you actually start off with so there's two main types of libraries that you can do for the MinION. If you have amplicons, you want this type of library because you've already got double-stranded amplicons, and really what you want to do is just stick on these adapters onto the end, so starting at the step where the arrow is, and then create these products which can then be loaded onto the sequencer, which contain the motor on the adapter. The motor I talked about earlier when I was describing the flow cell. The motor protein sits on the end of the adapter, which is ligated onto the amplicons, which mean that when you put them into the flow cell, they are able to interact with the pore and initiate that sequencing read. There's another type of library preparation method, which we call the rapid method, and that uses an enzyme called a transposase, which can be primed with adapters, which means that you can enzymatically cut into a double-stranded piece of DNA and insert the adapters. And this is very useful for sequencing genomic DNA, but for PCR products, you don't want to cut into the middle of your product. You want to sequence the whole thing from end to end. So we use the ligation chemistry on the left. So the final piece of the puzzle is the data analysis that we need to be able to do. So when I was we generated this data for these Ebola samples that we had, and we needed to be able to actually do the analysis, and that involved doing the base calling, which I talked about earlier, but it also comparing to other genomes and generating a phylogenetic tree. So we needed to be able to upload this data because at the time there was no such thing as a base caller that could run on your laptop that you had with you. So we needed to be able to use what was what was the only base caller available back at this time in 2015 was a cloud-based caller called Metricore. So we needed an internet connection to be able to upload the data to the cloud-based caller. So initially, I took the laptop back to the hotel I was staying at, which is pictured in the top left, and I uploaded the data using the hotel's wireless network, which was probably one of the faster connections in the city. So that was a satellite connection. We also investigated how expensive it would be to have our own satellite connection. The answer is very expensive. And then we eventually settled on using just a typical 3G hotspot, like the ones shown here. And this one was given to me by the World Health Organization, who organized the trip to Guinea. And I actually went around, bought some SIM cards of people in the street and top-up cards, and tested the different speeds. And we eventually found this network where we could get a fast enough upload speed such that we could upload the data from one Ebola genome in a reasonable time frame of about an hour. So that was what we did going forward. So as I said, back in 2015, the only available base caller was a base caller that ran in the cloud called Metricore. Now there's many more options available to us, including local base calling on the laptop, your laptop itself, and another local base caller called Albacore. So things have moved on since we did this, and are making this type of thing easier. So Nanopore, as I mentioned earlier, because it's a single molecule, it's measuring the current from a single pore, single DNA strand moving through a pore, is what we call a noisy or aeroprone technology, and that terminology basically comes from the distinction between amplifying a single molecule to create clonal copies of it before you sequence it, which gives you amplifying the signal, or sequencing just a single copy, where you're more likely to make errors in the sequencing because you have much lower signal to noise. So we in collaboration with a guy called Jared Simpson, who's at the Ontario Institute for Cancer Research, developed some tools for doing read correction, and we were able to apply this to Ebola to be able to call the SNPs, or the single nucleotide polymorphisms, the single changes in the genome which we use to do this type of analysis, very accurately. And we actually benchmarked that against the gold standard and found that it was able to call all of the variants that we had in the genome. So that was possible by taking the base called sequences and then combining that with the original Nanopore emission data, so the raw data, so including both the base calls and the raw data to increase the accuracy of the variant calls. So this is an analogous application, but for example, sequencing a bacterial genome using this algorithm, the accuracy that you can get from using the raw data as well is 99.96 at 300 times coverage. And this is actually an alignment of Ebola sequencing reads, whereby you can see the single sort of sporadic chain differences in the sequences, which are actually errors, or sequencing errors, which are labeled on there, and then a genuine biological change between the sample that we've sequenced and the reference genome, which is actually a real SNP. And you can see it's quite easy to, in this case, quite easy to see that that's a real SNP because it's supported by the majority of reads. But in certain circumstances, it's very important to use the raw data to improve the accuracy of these calls. So we came up with this big long pipeline, bioinformatics pipeline, which has now been actually Dockerized, and we can run this on a laptop right there in the field. So things have really moved on a lot since then. But it's important to design your analysis because it's properly, because MinION data is very different from other types of data, so it's necessarily complicated in this case. So we also had a question, a research question when we were doing this, which was basically, can we set up this lab and can we generate genome data in the field in a useful time frame? And I think that that was certainly proved to be very successful, more successful than maybe we even expected. And this was going. This is an image of a funeral going on during the Ebola outbreak. It's an incredibly tragic outbreak, more than 10,000 deaths. But one of the main drivers of the epidemic was unsafe burials, and in this case, you can see the protective equipment is being used to safely bury this victim. But as I said, one of the main drivers of the actual epidemic was unsafe burials, and the WHO, who produce during ongoing epidemics, they produce what's known as a situation report. And this is an excerpt from the situation report in October 2015, where they say that, however, he is not a known contact of a previous case, and the genomic analysis suggests he was not infected with the strain of Ebola virus responsible for the most recent cases in Conakry and Furikaria. And this is the first time that genomic analysis had ever been included in a situation report, and this was data that we had provided to the response control in the previous week, and so that was very gratifying that we were able to do that. So this is the hospital, the main hospital in Conakry, in the capital city of Guinea, where we had the lab set up, and this is me on the left with Dr. Masuma and Miles Carroll setting up the first Ebola sequencing run in Guinea, and actually probably anywhere. And the first data that we got off the instrument, we were able to recover, hundreds of X coverage of the Ebola genome within as little as 30 minutes after starting the run. So this is a very rapid assay once you've actually done all the lab work up front. The sequencing component of it is very quick indeed. And the initial batch of genomes that we generated, I think it was 14, showed that the phylogenetic analysis of those genomes showed that they fell into two distinct clades. And this actually wasn't known at the time, because there was such a lag period between what was going on in the field and what was actually known about, due to the fact that if you don't have a mobile lab, the only way that you can get genomic data for these samples is by collecting them all up, packaging them and shipping them out with a special courier which is happy to handle potentially hazardous RNA, and then send them to a lab which is equipped to handle them and do the genome sequencing there. That whole process has got a lot of cost and a lot of time associated with it. Whereas if you can do this in the field, you can avoid all of those bureaucratic hurdles and the time taken, which means that you can get the data in a much quicker time frame, and you might actually be able to do something about it. So by the time we got to the end of this project, the lab had been upgraded to its own porter cabin. These are some of the people that staffed the lab, did the lab work, including Joseph Bore, a local Guinean scientist who we trained, and you can see him working away, making libraries for the MinION. So we had a PCR area, a sequencing area, and that's the DNA library being loaded into the flow cell. And just to show how, just to illustrate how remote the actual sequencing was, you can see this picture from Lauren Cowley, who was working in the lab, has taken directly out the window looking into dense jungle. So you can see that this was genuinely right in the field, and it was enabled by the 3G connection to upload the data, which I mentioned earlier. So, look at these are some overall stats of the number of cases in Guinea from the date that we arrived in country to the final case of the outbreak. So we arrived in March 2015 when there was 265 cases, of which we sequenced 13 of them. This was near the end of the outbreak, and the numbers of overall cases were dropping. But during that time, the numbers that we actually sequenced increased as we got the lab more established to a period of time in June or July where we were pretty much sequencing all the cases that we wanted to. The ones that we didn't do were really cases where it wasn't necessary to sequence all of the isolates, for example, in family groups and things like that. And the graph on the bottom right shows the time between when the sample was first collected, so that's basically when the blood was drawn, and the date in which the sequencing data was available. And as you can see here, for a number of these samples, that was only 1 or 2 days after the sample was collected. And that just goes to show how powerful mobile labs can be when you consider that the alternative would be that it would be sent back to a lab with a conventional sequencing instrument in, and it might take months before you actually have the data. So that's that really shows you that the power of a mobile lab can generate data quickly, and it can generate it in a time frame where you can actually still do something about it. So the phylogenetic analysis, which is effectively a tree illustrating the evolution of the virus over time. The two boxes, which are the two boxes there, the dotted boxes, illustrate the two clades that I talked about earlier, the two lineages which were circulating in the country, and then these are zoomed in versions of that, showing the evolution of those two lineages over time, and the color of the dot tells you which part of the country or which prefecture that sample came from. So you can see that the virus was moving from one prefecture to the next, to the next, to the next, and that's driven by movement of people, infected people that are transmitting the virus from one geographic location to the next. And the way that you can reconstruct this tree is that as the virus amplifies, errors are introduced into the genome, and those errors, if it's transmitted, persist, which means that you see a change in the genome which wasn't present in the previous isolate. So if you compare the genomes using these types of methods, phylogenetic methods, you can create a tree which shows how each sample links to previous known samples. And that can be very useful information if you're tackling an outbreak, because you're able to see across big geographic regions or even countries which isolates are related to which isolates, and that might not be so obvious if you're dealing with this on the ground with a sort of very narrow focus. The people are quite mobile, and they're able to move the virus across large distances, especially if they're using air travel. So this kind of analysis is very important for being able to see how cases relate to each other, and that was very useful information for the people on the ground. So this plot here is an example of how the genome data, the tree in the top left, compares to a more conventional epidemiological picture that you might draw up if you were doing this sort of research by doing interviews and talking to people and doing this sort of thing. And in this case, the genome data exactly recapitulated the story which was developed by epidemiologists. So this is to illustrate that this type of thing could become an asset, or maybe even a replacement for epidemiologists working on the ground, doing a lot of laborious manual work. So this work was all published by our group, along with nearly 100 collaborators as a Nature letter, as the first time that portable genome sequencing had been used for surveillance of an epidemic. And these are some of the press stories that were written on the article. This is a UK paper called The Guardian, The Verge, and even Bill Gates picked up on this story. He's obviously his charity is very interested. His foundation is very interested in technology which can be applied to the developing world to reduce the burden of disease. So this is the type of thing that the foundation would be interested in. So I'm going to have to increase in speed because I've only talked about Ebola so far, and I've still got quite a lot to talk about in Zika. So we had a grant that was funded by the Medical Research Council as a rapid response grant to use what we'd learned about doing mobile genome sequencing for tackling Ebola for doing Zika. And both of these viruses are made headlines, and they're very worrying outbreaks. The Ebola, because it's a very very high mortality, scary virus, and equally Zika was very worrying to people because of its link to birth defects. So but there's a big difference in the way that these viruses travel. The Ebola is a zoonotic virus, assumed to be a zoonotic virus, and which means it comes from an animal reservoir and then is spread person to person. Zika is a virus which is spread by mosquitoes. So the mosquito bites someone who's got the virus and then is able to spread it to other people. So it's not exactly the same. It's not exactly the same application, but nevertheless, genome sequencing can still be used to understand the structure of the virus of the population of viruses that are circulating, and that can tell you quite a lot, as I'll come on to in a minute. So the question that we had for Zika surveillance sequencing was really what is the burden of Zika that is circulating in Brazil. This is we're only working in Brazil, although Zika is obviously right across the Americas, and we wanted to try and sequence a very large number of genomes. In the previous in the previous project with the Ebola, we were sequencing sample by sample by sample, which was quite a laborious process. And what we wanted to try and do is to multiplex multiple samples on one run. And by that I mean prepare a library which contained multiple samples, but where the reads were identifiable by a small molecular barcode, so that we could increase the throughput of the sequencing lab. So we wanted to do 12 per flow cell, so it's around 12 per day if we possibly could. And this was the team that we assembled. It's a mixture of Brazilian, British, Portuguese, American scientists, and we had 4 teams that we were working in. We actually travelled over 1,000 kilometres from Recife to Salvador, from Natal actually to Salvador, and in the picture there you can see Jacqueline, who is a Brazilian student who we actually now have working in our lab at this very moment. She's come over to do a project, and she's working on improvements to our Zika sequencing protocol. But we did this journey by bus, and we stopped in the central public health labs of the federal states of all of these places. And when we got there, we pulled out the metadata, the samples, and we attempted to sequence them. You can see doing RNA extraction, PCR amplification, genome sequencing going on in the lab. In addition to that, we had a team of entomologists that were looking at the capturing mosquitoes so that they could understand the burden of each individual species in each location, and see if we could correlate that with either the number of Zika positives that we found or the genomes. And this is, they're speciating the mosquitoes under a microscope there. And this is a animation showing where the ZiBRA bus went along the northeast coastline. And in each place we screened hundreds of samples which we were given by the public health labs, and in each there was on average about 10% that were positive for Zika. So it's so there was quite an attritional process, because we still had to extract, organise and test every sample, even though we only got 1 in 10 that were positive for Zika. And then, in addition, the group went out collecting mosquitoes, and they found multiple species of mosquito in each place. So these ones are the, I think this photo is the Aedes aegypti, which is the species which actually carry Zika, and that was the one that we were particularly interested in. And you can see from this heat map here that we collected the highest number of Aegypti in the Recife and Maceió states. And from testing the samples, we had a number of positives. The largest number of positives was from the Recife. We also had others from Natal, João Pessoa, Maceió, and Salvador. So this is the number of samples. We made 55. We attempted sequencing 55 samples in a week over 5 runs, and we ran into some severe technical difficulties, and that is because the abundance of Zika virus in human blood is incredibly small numbers of viruses. So this is a plot showing this is a real-time PCR. There's something called the cycle threshold value, which tells you the number of it's a proxy value for the number of starting molecules you had in your sample. And for the case of Zika, it was 36 was the most common value for the samples that we tested. So that tells you that there was a very small number of starting molecules in the sample, and that was making the genome sequencing very difficult. We were only able to properly sequence these 8 samples. If you define properly as being more than 50%, really we would like to have been sequencing the entire genome. So we had some real problems with that. We weren't the only ones. This is another group, Christian Andersen's group at Scripps in San Diego. They were also seeing similar problems. So we ended up designing a website which I wrote called Primal Scheme, and we had the interface was made by Andrew Smith, and this basically is a piece of software for designing primers for a viral genome, where the primers can be used in multiplex. So the primers can be included many reactions in the same individual tube, and the way that we did this was by choosing very high and very tightly controlled binding temperatures for the primers, and that meant that we were able to amplify the entire genome using only two reactions. And once we implemented this new method, we were really away. And this is an example of some data from Christian Andersen's lab, which I just mentioned, where they tested this new method, and basically we were seeing pretty well the whole genome in that being able to be sequenced from those 2 reactions. So so we went back to Brazil and had another attempt, and this time, as you can see from the table at the bottom, we were able to achieve genome coverages in the 90s for a number of these samples. So we had eventually we were able to generate the data that we needed, despite the technical challenges that are associated with Zika. And this gave the phylogeneticists plenty to work with, and they produced this tree here showing that there was multiple introductions of Zika in Brazil, that multiple clades were currently circulating. You can see a clade associated with southeast Brazil, northeast Brazil, another one of northeast Brazil. So very diverse viruses able to be detected. And what we were trying to do is they were trying to use this data to calculate what the likely introduction date into Brazil was. And if you use some advanced modeling methods, they were able to calculate that the likely introduction dates for these different regions, South America, Central America, and Caribbean, were as shown on these plots here. And this tells you that Zika had been circulating unknown, or that cryptically, as they call it, for a very long time before the national health authorities realised what a big problem they had. And that really tells you one of the sort of key important is that can illustrate really one of the key elements of genome surveillance, is really knowing what you've got a lot sooner, and it could have potentially been controlled if it was known about earlier. The problem was that the symptoms were quite similar to some other viruses which were endemic in the country, like dengue and chikungunya. So it was flying under the radar for a period of time. So in conclusion, I've tried to cover as best I can some of the key elements of the development that we had to do to be able to bring a mobile sequencing lab both to West Africa and to Brazil. And I've talked about some of the technical challenges which we had to overcome. in order to do that, particularly for Zika, which was a challenging thing to sequence. But I hope that you'll take away some of the importance of genome surveillance and what it can do, because we are becoming an ever more densely populated globe, with ever more air travel. So these are sort of conditions which are perfect for the spread of viruses, and we need more tools in our arsenal so that we can control these things when they arise. So thanks very much for listening, and I'll take any questions now.
We will now start the live Q&A. Portion of the webinar. If you have any questions you'd like to ask, please do so now. Just type in the green Q&A button at the lower left of the presentation window. Type your questions into the box that appear on the screen and click on the send button. We'll answer as many questions as we have time for. So let's start with our first audience member questions. What future developments do you anticipate for mobile labs?
So we are attempting to develop and improve the current methods that we have. We are working on all elements of the technology, really, and we've just been funded for a further 5 years to further develop the lab. One of the key things that we're trying to do is to improve the speed at which it takes to go from sample to answer. So that really involves taking a sample, a clinical sample, and making it safe. So we need to find ways of isolating RNA or DNA from a sample in a safe environment, which means inactivating virus. It means that you have to do it in an environment which is safe for the actual operator who's actually doing the extraction. So we use flexible film isolators, things like that we can do. We can manipulate and handle samples in a safe environment, but that can also be transported, because these things are normally fixed in very large sorts of specialized labs. So once we've got the RNA, we're then working on, or DNA, we're then working on ways that we can improve the speed at which we can go from a sample to a sequencing library. At the moment, that includes using PCR to amplify a very small number of molecules that's in the sample to a larger number, so that they can be used for sequencing. But these are still fairly typical molecular biology steps with enzymatic reactions and cleanups and more enzymatic reactions. So we're trying to streamline that whole process, maybe even automate it onto portable microfluidic devices. And we think we can get this down to the sort of hour. At the moment, it definitely takes a whole day. We're hoping that we can get this down to sort of 1 or 2 hours for sample to answer. So certainly the speed is one thing, and I think the cost. The more samples that you can run at once, the effective cost goes down per sample, and it's in order to develop something which is going to be widely used, it needs to be cost effective, and especially if we're going to be using it in the developing world, where the cost of these things is extremely important. I think the MinION itself is a big step forward in that respect, because the device itself has a very low value, and you're only paying for the consumables, as opposed to having to pay for the instrument as well.
Josh, we have a really captivating audience today, and our next question, and we have time for one more, is really interesting. If there was an outbreak of an unknown virus tomorrow, what would you do?
A lot of what I've been talking about today is really revolves around surveillance, and that sort of implies that you know that something is there. If and you're really keeping an eye on it. If there was an outbreak of an unknown virus, you know, we would have to use some different methods in order to try and identify what that virus was as quickly as possible. Now, you can still use DNA sequencing technology for that, but it needs to be you'd have to do a different method. You can't use PCR the way that we have to amplify the virus genome if you don't know what the virus genome is to start with. So there's an application area called metagenomics, which basically means all of the genomes in a particular sample, all the nucleic acid in a particular sample. And what we would do is take all the RNA or DNA in a sample and sequence it en masse and try to identify what we thought using databases of existing known viruses, what was in that sample. And then if we could identify what it was in that way, we could then switch to the surveillance mode where we could design an assay which was specific for that virus, and it would switch back to the surveillance mode, like we've just been talking about. But certainly we would expect to be able to do these things rapidly, and we have the equipment and the facilities ready to deploy this at the drop of a hat.
Thank you, Josh, for that informative presentation, and thank you for your important research. We just want to also thank LabRoots, and we also want to thank our sponsor, Oxford Nanopore Technologies, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us, and we hope to see you again soon. Bye-bye.